I suffer from short-term memory loss.
Yes! That's exactly what you say.
1, 2, 3, 4. I like sand. Sand is squishy. Mommy, can I go play with them?
So we're going to talk about the mystery of missing heritability. So forget-me-not disorder is found in a small school of fish, and it causes those affected to have little to no short-term memory. Forget-me-not is more commonly seen among blue tangs raised in captivity versus in the wild. The disorder is expected to be 82% heritable, but a recent genome-wide association study, or GWAS, which assumes an additive model, has identified 24 signals, or SNPs, accounting for 64% of the heritability. So right there we can see that not all of it has been accounted for in the GWAS. So, answer the following questions. A. What does it mean that the disease is 82% heritable? B. Suggest one possible reason the statistics might account for missing heritability and justify your answer. And C. Assuming the GWAS had sufficient power, please provide 3 sources of missing heritability and explain each. So we're going to break down each of these parts and explain a correct answer and also the significance. So for A, what does it mean that the disease is 82% heritable? This just means that 82% of the variation in the phenotypic expression of the trait, in this case the disease, forget-me-not, arises from the genome. This is negating any environmental factors or other things. It's the nature aspect of the nature versus nurture debate. Now B, one possible reason the statistics might account for the missing heritability is that it says in the problem that forget-me-not disorder was found in a small school of fish. The small sample size means that the study likely lacked the power needed to detect the sources of heritability, such as rare variants or common SNPs with small effects. A small statistical power means you're not going to detect everything in your assay in the GWAS, meaning that could be at least part of the missing heritability. And C, the longest part. Assuming the GWAS had sufficient power, just this statement alone lets you know that you're not going to be talking about the statistical measures or the lack of power, because it says that assume you had enough power, so the small sample size wouldn't matter in this case. So 3 possible sources of missing heritability. One is a gene-environment interaction. There might be gene-environment interactions that aren't accounted for in the GWAS. Because the disorder is more commonly seen in blue tangs sampled from captivity versus the wild, it's indicative that environment plays a role in the manifestation of the disorder. Another possible source of missing heritability is that some of the SNPs may be multiplicative instead of additive, which was assumed by the GWAS. This would mean that the effect of the SNPs would be underestimated using the additive model when compared to the actual effect of the multiplicative interactions. And then C, another possible source of missing heritability is that there might be gene-gene interactions. This is because the GWAS isn't looking for gene-gene interactions, it's just analyzing and detecting the individual SNPs, which it detected 24 of. So although the GWAS has the power to detect rare variants and other typical suspects for missing heritability, it's not able to detect gene-gene interactions. If 2 or more of the genes are interacting to produce the affected phenotype, the heritability of the disorder may be complex than the effects of the 24 individual SNPs. Understanding heritability is important when trying to account for the missing heritability we find. A lot of this might be because we don't have the statistical power currently in our GWAS studies to detect these rare variants or common SNPs of small effects. There might be gene-environment interactions which complicates what we're seeing. There also might be gene-gene interactions that GWAS currently doesn't look for. Also, a new line of study in genetics is testing different SNPs to see if they're really additive. Currently, genetics assumes that the relationship is additive, where one gene plus another gives the total effect. But a current line of study is testing, well, is it gene A or allele A times gene B or allele B to see if they square together and multiply, or if it is the traditional additive model. Forget-me-not disorder has a heritability of 82%. In an ideal scenario, we would be able to match this percent heritability exactly using only genetics. But in reality, things have become more complex, hence the problem of missing heritability. We can only account for 64% of the variation. Understanding where their missing heritability lies can lead to better prevention, diagnosis, and treatment of disease, and also can help design effective drugs, create individualized therapies, and better understand how to help people suffering from diseases and disorders.
keep swimming, just keep swimming, swimming, swimming. What do? We swim, swim.